Hi everyone, and welcome to the Biological Evidence Collection lecture. Let's get started. So I want to start with a quick review of some of the DNA information that you learned in Survey of Forensic Science. DNA is called deoxyribonucleic acid. It's composed of two chains that coil around one another, forming what is called a double helix. The helix chains are formed by alternating sugars and phosphates. Each sugar is attached to a base. The phosphate, sugar, and base combination are called nucleotides. Each nucleotide has a complementary pair across the DNA helix, with the connections between the bases holding the whole structure together. The bases always connect in matching pairs. Adenine pairs with thymine, and guanine pairs with cytosine. The bases will not connect to one another in any other way, so you won't get adenine connecting to cytosine, for example. The DNA that forensic scientists prefer to use is called nuclear DNA, or nDNA. As the name implies, nuclear DNA is found in the nucleus of the cell. Inside that nucleus are pairs of chromosomes. You can unwind the chromosome structure to release the DNA strands. Now, most people are born with 23 pairs of chromosomes, so they have 46 total chromosomes. Half of these chromosomes are inherited from your mother, and half are inherited from your father. All of your chromosomes are contained within a cell nucleus, usually 2 copies of each chromosome within each cell nucleus. In order to individualize, forensic biologists focus on small parts of DNA strands with high levels of variation, and these are called short tandem repeats, or STRs, and they're typically locations where 4 bases will repeat multiple times. While the sequence itself of the 4 pairs in the STR locations does not vary frequently, the primary highlight for forensic biologists is the variation in the number of repeats. The number of repeats in each STR location is affected by the number of repeats from both your father and your mother, creating a more individualized profile. With probabilities, the higher the number of improbable outcomes, the more unique the outcome becomes. So the value of STRs for forensics is not just the probability of the number of repeats in one particular section, but rather the probability of the number of repeats from multiple STR locations all matching up. While each has a more common occurrence, when all of the different probabilities are taken together, they provide a much stronger individualization. The FBI developed a DNA database using 13 different STR locations, noting that the probability of a match in all STR locations from unrelated, randomly mating populations of Caucasian Americans was 1.74 times 10 to the negative 15th, or about 1 in 575 trillion people. One of the most common methods for DNA replication is called the polymerase chain reaction, or PCR. This technique is a relatively simple process by which the DNA is unzipped and then replicated. First, the DNA is heated, which helps break down the double helix and unzips the chains. Think about taking a ladder apart by cutting down the middle of the rungs. The next phase is to rebuild each strand separately. New bases are added to the sample, and then they rebuild the ladder from each original half. So you started with one ladder, cut it down the middle into 2 pieces, and then rebuilt the full ladder into 2 different pieces using new material, leaving you with 2 full ladders. Doing this over and over and over creates a chain reaction in which DNA is unwound, rebuilt, then unwound again, and rebuilt, and so on and so forth. This repetitive process can help build up exponential copies of DNA relatively quickly. With this duplication, or what this duplication allows us to do, is to analyze smaller and smaller samples while still getting a full DNA sample for testing. In fact, DNA analysis has become so sensitive by the mid-2000s that we developed an analysis technique called touch DNA. Touch DNA involves the analysis of minute fragments of skin cells that are left behind when people touch victims, weapons, or anything else at a crime scene. Nowadays, we can get a full DNA profile from just 7 or 8 skin cells by themselves. The reason for that is the replication process. By dividing and recombining the DNA strands over and over, even 7 or 8 cells by themselves can produce enough usable material for a full DNA profile. So now that you've gotten that refresher on DNA specifically, let's talk more broadly about biological evidence. Biological evidence contains a broad spectrum of different evidence. In blood, the white blood cells contain nuclei that can be collected for DNA analysis. Sperm contained within semen samples also contains DNA as well. Bone marrow, bones, and teeth can yield DNA evidence as well, and they can be particularly helpful with identification of either long dead remains or highly damaged remains by things like fire or flood. Now, many of the other biological substances that analysts collect do not contain DNA themselves, but they may contain skin cells that provide nuclear DNA for analysis. For example, hair that's cut off of someone's head is not useful for nuclear DNA analysis because nDNA is located in the root of the hair. Similarly, saliva, mucus, and fecal matter do not contain DNA themselves, but they may contain some skin cells that accumulate as the substance exits our body, and so that allows for DNA analysis on them potentially. Given the variability of DNA availability in these type of samples, though, blood and semen are usually the most commonly collected substances that are sent for DNA analysis. So where do we find DNA evidence? Well, biological evidence can be present in a variety of locations, and that depends on the nature of the crime. Identifying where to look is crucial for proper evidence collection and case reconstruction. Biological evidence is often found on surfaces within a crime scene, including floors, walls, furniture, personal belongings. Blood spatter on a wall can indicate the force and the direction of an attack, while saliva on a cigarette butt may provide DNA that links the suspect to the scene. Bathrooms are another important location for biological evidence collection, as suspects may attempt to wash away biological evidence. Investigators should look for blood and body fluids in sinks, drains, or even between bathroom tiles. Now, the victim's body itself is also a crucial source of biological evidence. Blood, saliva, and tissue may be found on the skin, the clothing, or beneath the fingernails of a victim. In cases of sexual assault, swabs are taken from the mouth, genital areas, and other locations to recover potential DNA evidence. Bite marks may contain trace saliva, and bruising patterns can indicate the method of attack. Biological evidence like hair or fingernail scrapings can also be found under a victim's nails from defensive wounds. In addition to the victim's body itself, clothing from both the suspect and the victim can hold significant biological evidence. Bloodstains, semen, saliva, or trace DNA from skin contact can be recovered from fabric. Weapons that were used in violent crimes, like knives, guns, blunt objects, these can also be sources of identifiable biological evidence. Even if a weapon has been wiped clean, forensic techniques like luminol testing can reveal latent blood residue. In strangulation cases, ligatures like ropes or electrical cords may contain fibers and skin cells from both the victim and the suspect. Now, depending on the facts of the case, a vehicle may also provide a key location of biological evidence. Suspects can transport victims, dispose of bodies, or flee crime scenes in a car. Blood from an injured person, hair from physical contact, or even fingerprints mixed with sweat on a steering wheel can link individuals to a vehicle. Trunk linings, seats, and floor mats should be examined carefully for hidden biological evidence. And while electronic devices themselves don't contain biological evidence, they can hold DNA in the form of skin cells, like skin cells on keyboards, touchscreens, or earbuds. If a suspect was injured, blood or sweat may also be transferred onto a phone or computer. Forensic investigators take swabs from frequently touched areas like phones or computers in order to collect touch DNA, which of course can be used to link a person to a device used either during or after a crime. The key takeaway is that biological traces can exist in unexpected places, on weapons, in vehicles, on clothing, and even on digital devices. The proper identification and collection of this evidence can be an important piece of the puzzle, helping investigators to identify what happened and who was involved in the criminal offense. Now, identifying biological evidence at a crime scene isn't always as simple as spotting a visible bloodstain. Many types of biological evidence, like saliva, semen, or touch DNA, are invisible to the naked eye. Forensic investigators use specialized detection methods to locate and confirm the presence of these substances. Let's focus on three of the most common methods used in crime scene investigations. The first is ALS, or alternate light sources, and we've talked about this before. ALS is an essential tool for detecting biological stains, including blood, semen, saliva, and urine. things that may not be visible under normal lighting conditions. By using colored goggles or filters, forensic experts can isolate and enhance the visibility of stains on clothing, furniture, or walls. This method is particularly useful in sexual assault cases, where semen stains may be hidden on dark fabrics or in environments where body fluids have already dried. While the visual light search can be done anywhere in the scene, the investigators should focus on areas that were identified as relevant by the victim, the witnesses, or the suspect in the case. This will help to avoid collection of non-relevant samples. For example, consider a rape that occurs in a hotel room. As you've probably seen on TV, using ALS in a hotel room can provide you with way too many bodily fluid stains, more than you want, and definitely a lot more than are necessarily relevant to the events in question. By focusing the search for biological fluids on the specific areas of the room in which the victim reported having sexual contact with the offender, investigators can reduce the number of non-relevant samples that they collect and things that require analysis. Presumptive tests are another way to identify biological material, and these are typically fast, field-ready tests that use chemical reactions to indicate whether a suspected stain is biological in nature. These tests allow forensic investigators to conduct on-the-spot analysis before they send samples to a laboratory. There are a number of presumptive tests that can be done in the field or in the lab to identify if a sample is biological. The type of test is going to depend on the biological substance that's being tested for. Hemosticks and Kastle-Meyer tests are the two most popular tests when testing for blood. Hemosticks turn a green to blue-green color to indicate a presumptive positive for blood, and the Kastle-Meyer test, which is shown on this slide, turns a bright pink color when the solution comes into contact with blood. There are also specialized presumptive tests for semen and other biological substances. The thing to keep in mind is that these are presumptive tests only, meaning that they have a higher than desired rate of error. Most concerning are false positives, where something shows up as a biological fluid that it's actually not. This is why taking samples for further confirmatory samples at the laboratory is paramount to ensure that error is minimized. And that brings me to my last one, which is confirmatory tests. Now, these are laboratory-based procedures that provide definitive proof of the presence and the identity of biological evidence. These tests can distinguish human blood from animal blood, they can verify the presence of specific bodily fluids, and they can enable forensic scientists to extract DNA for profiling. Just like presumptive tests, confirmatory tests are typically specific to one or more type of biological fluid. For example, RSID, the Rapid Stain Identification Test, can confirm whether a stain contains semen, saliva, or urine, and a confirmatory test called HemaTrace specifically detects human blood. The reason that confirmatory tests are typically done in a forensic lab is that they are often either more expensive or more time-consuming than the presumptive field tests. For that reason, confirmatory tests are not often carried out at the crime scene itself, so you're more likely to encounter ALS and presumptive tests at the scene. All right, well, that concludes the biological evidence collection lecture. Thanks for watching, everyone.